Hi, I'm Alexis, and today for my Tour of the Cell project, I'm doing the interaction between a neutrophil and a Staphylococcus aureus. So I'll show you guys here. And just to explain the diagram, so this is as to scale as I could get it, but this is our neutrophil, and then right over here is our Staph aureus, so significantly smaller, almost 10 times smaller. And then this is a larger version just to show like the different surface proteins and a little about what's inside of the Staph aureus. So I just wanted to explain how neutrophils interact with Staph aureus and why this interaction is important for our body's first line of defense. Staph aureus is a gram-positive bacterium. It normally lives pretty harmlessly on our skin and in our noses sometimes even, but it does enter the body sometimes through a cut or a break in the skin, and it can quickly cause infection. So when this happens, our neutrophils, they're the most abundant type of white blood cell, but they respond pretty quickly and they try to find, engulf, and destroy that bacteria. So neutrophils do have surface receptors, kind of like how I was showing you for the Staph aureus, and you can see them here. And I also do have a breakdown on the side, and I'll include this as well. But these surface receptors will send signal during infection, and once invaded, neutrophils will kind of migrate to the site of infection. And once they do arrive at that site of infection, the neutrophils can attach to that bacteria through those receptors. And the attachment is more efficient when those antibodies and complement proteins coat the bacterial surface as well. So this kind of helps the neutrophil and the Staph aureus target each other, kind of see each other a little bit more. Once this attachment does occur, the neutrophil engulfs the bacterium through a process called phagocytosis. The bacteria will become sort of trapped. I tried to show this as best I could, but it kind of engulfs it a little bit, and the connection will eventually form, but this is sort of how it starts. And the bacteria, they become trapped inside this membrane-bound compartment. And if you look here, there is a phagosome, and that phagosome will fuse with lysosomes and antimicrobial granules, which you can see here, those granules. And those will be inside the neutrophil. All of this will occur inside the neutrophil. But inside that compartment, enzymes and reactive oxygen species will kind of work together, and it will cause damage to the bacteria's membrane, its proteins, and the DNA as well, which will ultimately destroy the bacteria. So neutrophils also have another defense strategy. They can release what's called nets. So they're neutrophil extracellular traps. And they're kind of like web-like structures, and they're made of DNA and protein, and it traps and kills bacteria outside the cell, and it prevents them from spreading as well. So Staph aureus does have a way around this, though. They do evolve, and they resist neutrophil killing sometimes. They have a thick cell wall. It makes it harder to break down. They also can produce antioxidant enzymes that neutralize harmful reactive oxygen molecules, which is ultimately what the neutrophil is going to use to try to kill them. And some strains of the Staph aureus will even release toxins that damage neutrophils, and they can temporarily survive inside of them. So this whole interaction does kind of highlight the differences structurally between prokaryotic and eukaryotic cells. Neutrophils contain membrane-bound organelles, like you can see here, with like a nucleus, and it does allow them to kind of organize the complex microbial system or antimicrobial system. But in contrast, the bacterium does lack all those organelles, and it relies on a protective cell structure, like you see here with these three rings, to survive in those hostile conditions. So overall, the ongoing battle between neutrophils and Staph aureus demonstrate how our immune system rapidly detects and eliminates those microbes. And it does help prevent infection. So that is my project. I hope you enjoyed.